Can you see my screen?
Yes.
Okay.
We can see your screen. May I know who's recording this session?
You are recording and Dr. Ravindra is recording. Should I also record?
That should be fine. Yeah.
So should I also
Yeah,
record?
you can also.
Okay.
You can also. Can you put your screen into full screen mode?
It's now full screen.
Presentation mode.
It is in presentation. Is it not coming?
Whatever you're seeing in the screen, the same thing we are able to see. It is preview. What is that called?
Sylvia, are you using two monitors?
Yes.
Please change the display.
Okay, one sec.
You're sharing a wrong display.
God. Is it fine now?
Yes. Perfect.
Okay.
I guess we can start.
Yes. One second. Can't see the time. All right. Good morning, everyone. Welcome to webinar series, Introduction to Computational Drug Design, co-organized by Pharmacy Council of India and Schrödinger. I'm Shelby, I'll be the host today. So there will be two sessions today from 10 to 11 and from 11:15 to 12:15. So we will go ahead and start with the first session, Introduction to Computer-Aided Drug Design now.
Lightweight.
Hi. Can you see my screen?
Yes, we can see your screen.
Yes.
Good morning, all. I'm Pritesh Bhatt. I'm a senior scientist with Schrodinger, and today with our first presentation, I would be discussing on introduction to drug design. So here in this presentation, we would be discussing all the background. So what is meant by drug design? So how does, in a very simple way, we will try to discuss on how the computational tools would help. All the detailed discussion we will be having in the coming presentation. So I request if you have any questions or any doubts, you can type in the chat box in the YouTube chat. So we can take up your question after our presentation. And the panelists, I request you to mute yourself so that we don't get disturbance in between. Right, thank you. Great. Okay, so let me start with the presentation. to begin with, let me give a brief description about the Schrödinger software suite. So since we are co-hosting, I would be happy to give a brief description about Schrödinger software, what all things we have here, then we will go into the actual discussion topic. So we have PyMol. I'm sure you might have used PyMol interface. This is a beautiful graphical visualizer. Many people use this for visualizing proteins or small molecule or a complex. So currently it is developed by Schrödinger. We have biologic suites. So here you have all the tools for predicting the protein's properties, like you can design vaccines, you have tools for designing antibodies, or you can understand protein-protein interaction, a lot of protein engineering tools like alanine scanning or cysteine mutation or protein designing tools, or even liability prediction tools like protein aggregation or hotspot identification. All such tools we have in biologics module. Then we have small molecule drug discovery suite, where we have all the tools which will help in designing small organic molecules. So why they are small? Because the molecule we are designing will have molecular weight less than 500. So here we have tools like docking, then homology modeling, pharmacophore, QSAR, free energy perturbation, hydration site thermodynamic property. So these type of various tools we will be having. Then we also have material science suite. So this platform supports predicting properties of the material. So it could be optoelectronic property, or it could be physical property or chemical properties, right? So many of the polymer companies use it. Petrochemical industries use these type of tools. Even even the pharmaceutical formulation, a lot of pharma companies use these tools for their research. So it could be to check the physical stability of the formulation, or how the nanoparticle forms, or what type of excipients would be really suitable. So all these sort of studies can be done using material science suite. So this is a brief description. In detail, we will be going on each and every topic in the coming presentations. Okay, so let us first understand what is meant by drug design. Okay, so before we go into the computational tools for drug design, let us try to understand what is meant by drug design. So why do you need a drug? That is to treat a disease. So what is the cause for the disease? So what is the root cause? What is that thing which is going abnormal during the disease, which makes you feel uncomfortable, right? So it is the protein which maintain the homeostasis in the body. So there are several pathways involved, right? Let's say if you are going for cell wall synthesis, or it could be cell synthesis itself, or it could be digestion. So you might have come across a TCA cycle. There are 12 enzymes in that. So it is all the proteins which are involved in body homeostasis. These maintain the normal functioning of the body. So there would be something going wrong with these protein. So what type of things going wrong with the protein here? Either there would be increased activity or decreased activity. That is the main reason for disease. And what is the cause, or why there is a overactivity or underactivity for the protein? So there could be two main reasons. The first thing is there could be increased or decreased quantity of the enzyme available in the cell. Okay, or it could be because of the number of molecules are same, but inherent property, inherent catalytic activity has got disturbed. So either there is too much increase in the output, or there might be too much decrease in the output, and that is because of mutation. So how mutation is causing this? So during the mutation, there would be change in the amino acid residue, and these amino acid residues are synthesized from genetic code. So if there is a change in the genetic code, then the protein or the amino acid residue may get substituted, and because of that, the catalytic activity may get affected. Okay, so this concludes it is the problem with the protein which we have to take care. Now let us try to understand something about the protein. So I'm just brushing up your knowledge here, trying to connect the things so that you would be able to understand things properly. Okay. Protein is made up of amino acid residues. There are 20 types of amino acid residues, right? So you can, there are different classes. You can classify them into different categories based on their charge, or the polar or non-polar nature. So there are positively charged residues, there are negatively charged amino acid residues, there are polar residues, there are hydrophobic residues, there are aromatic residues, right? So these amino acid residues polymerize to form the protein. Okay, so again, when we talk about structural aspect of the protein, so we can classify them into primary structure, secondary structure, tertiary structure, quaternary structure. Let us discuss. I have here a small graphical representation.
Amino acids is encoded by DNA. The amino acids are joined by peptide bonds, which link an amino group and a carboxyl group. A water molecule is released each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into two types of secondary structures stabilized by hydrogen bonds. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form beta sheets. Many proteins fold into a compact globular shape, with hydrophobic side chains sheltered inside away from the surrounding water. The functions of many proteins rely on this folded structure. For instance, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds oxygen. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body. Many proteins rely on the ability to recognize the shape of specific molecules in order to function correctly. The flexible arms of antibodies protect the body from disease by recognizing and binding to foreign molecules, and thus preventing the viral RNA or DNA to enter the cell. Collagen forms a strong triple helix that is used throughout the body for structural support. The calcium pump moves ions across cell membranes, allowing the synchronized contraction of muscle cells. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood sugar level. Alpha amylase is an enzyme with a catalytic site that begins the breakdown of carbohydrates in our saliva. Ferritin forms a hollow shell that stores iron from our food.
Right. So the tertiary structure or the quaternary structure of the protein is essential for exhibiting activity, right? So now we studied there are two types of protein exist in the body. So either it could be enzymes or it could be receptor. Okay, so let us try to understand the difference here. So enzymes will bring out the catalytic activity and the receptor would propagate the signaling. Okay, so we studied about the proteins. Protein exists in different form. We have studied like protein exists in the activated form and protein exists in the inactive form. Okay, so what is the difference between active form and inactive form? So this applies to both enzyme as well as the receptor. In simple word, I'll say protein can exist as activated form or inactive form. So the observation here is protein in the activated form is able to bring the catalysis, or it would be able to propagate the positive downstream signaling. But in the inactive form, it won't be able to bring any catalysis and the signaling will not get passed out. So what is the difference between these two forms, active form and inactive form? Is there amino acid residues changing? No, that is not happening here, because the primary structure or the amino acid sequence remains the same. So what is the difference in the protein happening which is causing it that we are observing difference in the activity? In one form it is able to bring the catalysis and in the other form it is not able to bring the catalysis. The main difference is change of shape. So like this you can see here. So either it could be in the enzyme or it could be in the receptor. Okay, in the enzyme, in one particular shape it is able to bring the catalysis, and in other shape the enzyme will not be able to bring any catalysis. And in case of receptor, if you consider, for example, here I have put a picture of GPCR, you can see the structural difference, or it could be ion channel, right? So how this shape change is triggered. So there is a very special molecule, we call it as substrate, right? Why it is special? Because it has two unique property, that is called as intrinsic activity and affinity. So substrate is that molecule which has very strong affinity with the protein. The moment you put it with the protein, it would go and get itself attached with the protein. So it has a very strong friendship. It likes to bind to the protein. Not only that, it has second unique property called as intrinsic activity. And because of this special property, now the substrate has a capacity to bring the conformational change in the protein. So you can see in graphical picture, when the substrate comes and binds, it would induce the conformational change or a shape change in the protein. Now with this shape change, in one shape, protein is able to bring the catalysis, and in other shape, protein is not able to bring the catalysis. Okay. Now let us come to the diseased state. Okay. In the disease condition, already there is either increase or decrease in the activity. Let us assume, let us imagine there is overactivity. Okay. Let us consider a disease like cancer, where there is cell overgrowth. Okay. So there is a particular enzyme which is overactive. So the moment substrate comes and binds, it would induce the conformational change, and that would trigger the positive response. So now what is the way to stop the activity of the protein? I have to suppress the overactivity of the protein. Now if substrate goes and binds to the protein, that would trigger the positive response. So if I prevent the substrate going and binding to the protein, then I can control, because substrate is that special property, has these two unique property. Any other molecule, any other organic molecule or phytochemical molecule, it will not have the intrinsic activity. It will have only binding affinity. So I should be designing a molecule which should be having higher binding affinity than the substrate with the protein. I hope it is clear now, right? So we should be coming up with the design of an organic molecule which should be having higher friendship with the protein than the substrate itself, because substrate is already available in abundance in the body, right? So you would be taking medicine for a very short term, right? It would be either for 3 days or 7 days, and you are taking a small capsule or a tablet, maybe 5 mg, 10 mg tablet you are taking, or a capsule you are taking, and in that small concentration, our designed molecule should be able to preferentially go and bind to the protein. That means our molecule, if it exhibits higher binding affinity, then only it would be able to go and bind to the protein. Okay. Now, what is that driving force for the affinity? What brings out the special friendship? Okay, it is all because of non-bonded interaction. Why I am saying non-bonded interaction? Because there is no covalent bond formation there. It is only the non-bonded weak type of interaction. So the interactions could be like ionic interactions, or it could be dipole-dipole interaction, hydrogen bond, hydrophobic interaction, right? Electrostatic interactions. So there is no permanent covalent bond formation. So once the molecule goes and binds, it would exhibit activity for some time, then it would come out. Okay. So if our designed molecule exhibits higher number of non-bonded interaction, then it would preferentially go and get attached itself. So which type of interaction is really very important? So I listed out several of the non-bonded interactions here: hydrogen bond, hydrophobic interaction, pi-CADD, pi-STAT, ion-induced dipole-dipole interactions, or ionic interactions. So which one contributing really very high for the binding interaction? So this was one statistical analysis reported in a PLOS publication. You can see here, it says all the interactions are equally important. It is not the single type of interactions which drives the binding affinity. Okay, it is not just the hydrogen bond or not just the hydrophobic interaction. It is all the types of interaction equally important, and they will have synergistic effect. Okay, so now let us try to understand here. We can try two of the approaches. If substrate goes and binds to the protein, then there is a positive signal, and then there is a deterioration. Already there is a disease happening, so it would get aggravated. So I have to make my organic molecule go and bind to the protein. So I can try out two approaches. The first approach is wherever the substrate is binding, okay, substrate is binding in this position, to the same position if I come up with a design of molecule which goes and binds to the place where the substrate is binding. Now since my designed molecule has a higher binding affinity, my molecule would preferentially go and bind to the protein. The substrate will not be able to come and bind. But our designed organic molecule will not have intrinsic activity. So conformational change it will not be able to bring in the protein. Protein will be stuck with one particular shape. And if protein gets stuck with one particular shape, it will lose the activity. Okay. So this is with this approach where the substrate is binding in the same place we are exploring our molecule to bind, then such approach is called as competitive inhibitor or competitive binding. Okay. Or otherwise, you can identify a different site than the substrate binding site. So you can make your molecule bind to some other site. And when this molecule goes and binds, now it will disrupt the shape of the binding site. You can see here. So the substrate doesn't have a space now to bind it because the shape has been disrupted. Okay. So this approach is called as non-competitive inhibitor, or it is called as allosteric inhibitor. So ultimately, whether it is a competitive inhibitor or an allosteric inhibitor, you are disturbing or you are blocking the conformational change happening in the protein, and that's how you suppress the activity. So especially in case of enzyme, if you prevent the conformational changes happening in the protein, that is more than sufficient. Then the protein will become dead, and we want to suppress the overactivity. So here in case of cancer, there was overactivity of enzyme. So when our organic molecule goes and binds, since it is now not able to bring out the conformational change, protein loses its activity and activity becomes normalized and the health condition improves. Okay. So if in case of receptor, now receptor signaling, receptor is involved in signal transduction. So here the shape of the protein is important. So the way in which you lock the protein shape, that type of signaling it will propagate. For example, you want to have an activator, you want to have a positive signal going downstream. So in such cases, you have to lock the protein in the activated form, active form. You come up with the design of molecule. When it goes and binds to the protein, see that protein gets locked in the activated form. If you want to inhibit that protein, then you make the molecule, you design the molecule such a way that it goes and binds to the protein in the inactive form, so that the shape will be locked in the inactive form and all the downstream signaling would be negative signaling. That is how you block the activity or you modulate the activity of a protein. Let us try to understand the different stages in the drug design. Okay, so it starts from target identification. So first I have to identify which is that protein which is causing problem. That is called as target identification. Okay, then you have to identify the moieties, chemical moieties, organic molecules, right? Then that stage is called as hit identification. Then you improve the potency, that is called as hit to lead, and then you take out all the problematic parts. So there might be toxicity, all those things you have to remove. That process is called as lead optimization. So once you get the optimized molecule, you have to test a lot of things in preclinical studies, in vivo, in vitro testing you have to done. So animal testing you have to do. So once you prove that molecule is really safe in animals, then you would approach FDA, right? So you will ask for the permission to test it in first time in humans. Okay. So all these stages before you try it in human, this is called as preclinical. Clinic is a hospital, right? So preclinical, where you are not going to hospital, it is in the animal house or in the research lab. So that is why it is called as preclinical stage. And then once FDA sees all the data you had generated based on the preclinical studies, then they will say, okay, this molecule looks safe, you can try it in humans. Okay, then you will start human trial, that is clinical trial. There would be 4 stages in that. So the first stage is phase 1, in very small population you would be trying it. Then phase 2, a little bit larger population you would be trying, and on a very large population you would be trying on phase 3. So once you finish the phase 3 trial, you can again apply to the FDA. You can say, we tested in humans, even there it is exhibiting very good activity and it is not showing any toxic effect. Then FDA will think about it, it will review the data, and then it will say, okay, now you can go for marketing, you can sell your product. Then the phase 4 trial starts, that is post-marketing surveillance. Okay, so it is really time consuming. So you can see here, it is almost taking 13 years, 13 to 15 years. So if you start today, the process of drug discovery, it would take minimum 12 to 13 years to finish, and it is very expensive, more than $2 billion US dollars you need. Okay, then how in corona cases people are finishing so quickly the trials, right? So if it is taking 13 years, corona is a very new disease, right? So it's a valid question. Now, in some exceptional case, FDA will give some exception. So accelerated process, they will say, okay? But it is still risky. So any toxicity studies, unless you test it for a longer time, you won't be able to identify that type of risk you have to take. But when you want to save somebody's life, that is when it is priority. They will take a calculated risk and they will give the approval. Okay, so that is why the corona research is really running fast, so people could finish quickly the preclinical studies, and they got the accelerated approvals. Okay, so why it is really taking so much time and so much money? Okay, if you see the molecule we are trying to identify as a drug molecule, if it just has the potency, that is not sufficient. The molecule should be really safe, right? It should not produce any toxicity. It should be having a decent ADME profile. What is meant by ADME profile? It should have decent absorption, distribution, metabolism, excretion, right? ADME profile. What does that mean? Molecules should be highly selective. Whatever target you are selecting, it should be going and binding to that target only. It should not be touching any other proteins. It should have decent solubility. It should have good bioavailability. It should have a decent clearance or a half-life, and it should be permeable across the membrane. And it should not have toxicity like CYP inhibition, hERG blockage. And moreover, the molecule should be synthetically feasible. So we are trying to identify that one molecule which should be having several properties in it. That is a big challenge, and that is the difficulty. So to identify such molecule having all the property in the acceptable range, you have to try and test several molecules. You have to try out, you have to synthesize, you have to test. Then only you would be able to identify that one molecule. So all those failed molecule cost would increase, right? So the cost has to be bared by that one molecule which becomes successful. So all the failed molecules, testing cost or synthesis cost, maintenance cost, all has to be bared by that one molecule. That is why it is really expensive. So there was a review reported, published in Nature Review. Okay, so per project, if you see, the number of molecules synthesized would be about 2,000 molecules. Okay, this is in the traditional method. People, if they follow traditional research methodology, about 2,000 molecules they will synthesize. Out of these 2,000 molecules, hardly 10% of molecule would exhibit activity in the desired concentration. That means in single-digit nanomole concentration. Okay, that means 10% of 2,000 would be about 200 molecules. Okay, now these 2,000 molecules which are exhibiting very high potency, if you test their druggability, okay, or ADME profile, it's only the 0.41% of the molecule would exhibit decent druggability in nature. So that means it is hardly out of 200 molecule, only one molecule which will exhibit a druggable profile. And that molecule reaching to clinical trial is only 34% chances. So you can understand. So out of 3 molecules, 2 molecules will fail in the clinical trial. That is the statistics they are saying. So it is a high risk game, and you can see up to even preclinical stage, if we go, about $800 million we have to spend. So is there a way you can cut short this? So is there a way directly I can identify most potent molecule or the molecule having decent ADME profile? Can I use a smart approach? And nowadays, FDA has increased stringency. The number of molecules getting approval has reduced tremendously. So because of lot of toxicity effects, or molecules are exhibiting late toxicity effects, the FDA is really very careful in giving approval for any of the molecule. You cannot get easy approval, right? So that is why again the challenge has increased. So here in such scenario, you can use computational tools. So you can run the simulation, you can generate a computational model, like an animal model, how you use in preclinical models, right? Preclinical trials. So here computational models you can use, and you can try to predict the property of a molecule. So in the various stages of drug discovery, you can use, okay, starting from, it could be genomic analysis. Okay, once you identify the target, then you can find out whether that target is really meaningful or not. Can I identify is that target, if the structure is not there, you can generate a structural model. You can check whether the target is meaningful for target validation. So a binding site, is it druggable or not? Can you dock the molecule? Is the pocket sufficient large or not? You can come up, you can use the computational tools for hit and lead discovery. So you can go for virtual screening, de novo designing, pharmacophore mapping, right? Then further, the lead molecule, you want to go for lead optimization, property improvisation, and toxicity removal. In such cases, you can use again various computational tools, QSAR, FEP, free energy perturbation, binding affinity calculation. So all these type of tools. So throughout in the preclinical trials, studies, you can use computational tools to predict the property. Okay. Now, drug discovery is a high risk game, right? You have to invest 2 billion. I showed you initially $2 billion you have to spend, and ultimately it is only 34% of the molecule which reach to the clinical trial becomes successful. But if that one molecule gets successful, you can imagine the amount of money you will get back. So you should be ready to take the risk, like here I have shown in the picture. If you jump to the cliff, then you get that excitement, right? Now, those days have gone. Now the technology is really improving. You need not really stand in queue to get a ticket. You need not even really go to a grocery shop to purchase any commodities or any things. In fact, you need not even go to a restaurant to have food. You can, sitting at home, you can order the food and it will be delivered at your doorstep. Technology is changing, right? So same way, in drug discovery, we have to go with the technology. We need to implement the fast technology where rather taking 13 years, can you come down? Can you finish your drug discovery preclinical stage in just like months? It could be 12 months or 18 months or 24 months. We have to change our approach. Even we have to think very smart, right? So here you saw the zebra, if it runs, then it would be slow. It has to take a bike. Now, if lion also follows the same technology, if it is not smart, if it doesn't become smart enough, it won't get food. So we have to use a smart approach, right? Fast approach, and that is the computational tools. So those days have gone where everything you test and try in lab. You go for high throughput screening, robotic arm would be picking the molecule, do the activity. So that is expensive, that is tremendous job. Now we are implementing the technology, we check, we run the simulation, we predict the property of molecule, whether the binding affinity is really good, whether the molecule has great potency or not, whether it is having any toxic profile, is it having any genotoxicity, liver toxicity, cardiotoxicity, all those toxicity profile you can predict. And with that, the molecule which are predicted really fantastic, with the very good property, only those molecules you select go for the synthesis. So with that, you can reduce the number of molecules synthesized. So this is the picture I showed you initially. So in a project, about 2000 molecules will be synthesized and only 10% will show good activity. That means 200 molecules. And out of that, one molecule will be druggable, and that becoming successful is only 34%. That is what the story I had shown you earlier. Now, if we use the computational tool, instead of just testing 2000 molecules, I can try out billions of molecules. Okay, I can use the computational models, I can use supercomputers. Now the technology has changed. In Schrödinger, we are part of COVID research consortium, where we have screened billions of molecules. If you take in reality, testing those molecules would have taken some 2000 years. That we finished just in 2.5 months, right? So you can screen billions of molecules, you can apply filters, predictive property filters, and you can cut down the number of molecule, right? The best molecule only you can select. So you can go for the design of best molecule in the computer. Only those molecules now which are exhibiting very high potency and devoid of any toxicity, only those molecules you can focus and you can synthesize. That's how you can really reduce the money to be spent on the research, the molecule to be synthesized, and that's how you can cut short the time. Okay. Do we have an example? Of course, there are several molecules now which are in the advanced stage or already marketed. So one of the story is our own story from Schrödinger. We have the molecule. This was in collaboration with the Nimbus Therapeutic. So this is the target here we explored was ACC, that is acetyl-CoA carboxylase. Here, just in 16 months, using computational approach, the whole project here was driven based on computational approach. So we went with the virtual screening. Okay, so this target was very old target. So from past 16-17 years, people have been working, and the project was not progressing beyond preclinical stage. So here we started computational approach. We started with the virtual screening, identified a molecule within 16 months. Okay, we could identify a clinical molecule. Currently, this molecule is in the clinical stage 3B, it is running. Okay, so using computational tools, you can really speed up the process. Okay, so we will be discussing in forthcoming discussion what all the different approaches present. In detail, we will be going. So basically, there are two approaches. So in computational drug discovery, we can either go with structure-based drug discovery. That means we will be using the structural information of the receptor. We will see the binding site, what type of interaction the receptor can offer, that we will check, and we will try to design a molecule complementing the binding site. That is called as structure-based drug design. Here, you need to have the structural information of the receptor. So crystal structure of the protein is essential here. So such approach is called as structure-based drug discovery. In many of the cases, you will not have the structural information of the target. Like for example, in case of ion channels or GPCRs, you know, this is the target, this is the protein which is causing problem, this is the protein which is responsible for the disease, but the structural information will not be available. Many people might have reported, so based on their trial and error approach, people would go for synthesis and they will report this molecule is less potent, this molecule is more potent, this molecule is very less active. So like that they'll. So all those ligands information we can use, and we can come up with the design of molecule. That approach is called as ligand-based drug discovery approach. So here you can go with the fingerprints or shape similarity or pharmacophore. In case of structure-based drug discovery, you would go with molecular docking or molecular dynamics, protein modeling, these type of approaches you would be following. So depending on the information available, okay, whether the what type of information available about the target and about the inhibitor, you would decide on what type of approach would be really most suitable. So should I go with structure-based approach or should I go with the ligand-based approach? To decide that, you can use this information. So here I have made 4 quadrangle. Okay, let us imagine I do not have the target which I am working is very new, like in corona situation, now in COVID situation. So this disease is a very recent disease. So there is no much information available in the beginning. So we don't have the structural information of the protein, or even the inhibitors are not reported. So in such cases, we have to explore. You have to go with combinatorial chemistry. In the lab, you have to test. So once you get the preliminary data, then you can use it for computational studies. Okay. Let us imagine I don't have the receptor structure, but only I have a lot of ligands reported. So people will just take, now let's say some phytochemical they collect and then they just test the activity. So now I have ligands information, ligands activity information I have. So if in such cases, I can go with the ligand-based drug discovery approaches. So I can go with either pharmacophore, QSAR, ligand similarity, these type of approaches I can use here. Let us say I have only the target structure known, but there is no information available. So in corona cases, we are having these type of situations a lot of time. So since it is new organism, so people are doing research and they are identifying new protein targets and they are crystallizing it. So structural information you are getting, but till now nobody has reported any inhibitor because it's a new protein, new target. So in such cases, you can go with the structure-based drug discovery approach where you can try virtual screening, de novo design, right? So these type of approaches you can go for, okay? If all the information about protein and inhibitor is there, so let's say it's a very old target, okay? So protein crystal structure is also there, binding site inhibition information is also available, and lot of inhibitors has been reported. So lot of research has happened. In such cases, we will have all the information. Then in such cases, you can go with the combinatorial approach. So you can start with ligand-based drug discovery approach where you can use where you can come up with the QSAR or shape-based search, or you can go with pharmacophore search. You can run a virtual screening. The best molecule you can go for a structure-based drug discovery tool where you can go with the structure-based virtual screening. The best molecule you can identify, you can go with free energy perturbation methods to accurately identify what is the binding affinity. So all the combination or hybrid approach you would be able to try. So various approaches like for target validation, you can use computational tools like protein modeling and like in homology modeling you can use, or for binding site identification, we have various tools where you can see whether the particular binding site is it druggable or not. You can check with molecular dynamics whether the protein structure predicted is stable or not. Those type of analysis you can do. Further, for hit identification and lead generation, okay, first time you want to identify particular chemical moiety, is it useful or not? In such cases, you can go with structure-based drug discovery where you can use something like high throughput virtual screening or docking in simple way. In case of ligand-based drug discovery, you can go with the different types of chemoinformatic analysis where you can go with the QSAR, or you can go with the pharmacophore-based screening or shape-based screening, fragment-based drug discovery methodology you can use. And further, for lead optimization, once you have a decent active molecule, you want to increase the potency and you want to remove the toxicity. In such cases, you can use various docking methodologies. So it could be induced fit docking or fragment-based docking methodology. It could be various scoring methodology for identifying the binding affinity, like MMGBSA, free energy perturbation, molecular dynamic simulations, right? So for designing new molecule, there are various approaches. Like in Schrödinger, we have tools like ligand designer, reaction-based enumeration. So if you design a molecule, what way you can synthesize that, what are the pathways can be explored, how many step reaction is really essential. And once you have the pathway, you can generate a library, you can enumerate the library. So further you can go with bioisosteric modification, various fragment based drug discovery methods. All these different type of tools you would be able to use. So here I will stop my discussion. So before I take up a few questions, let me share few information. So we have online certificate course. So if you are taking part in this seminar, in this drug discovery course, we will provide you the license on your machine. So for that you have to register. Okay, so first you have to register in this webinar series. So for that you can visit our web page. So in Schrödinger web page, in the event site, you can see the information. And further, you have to register in our in the Schrödinger website so that we can help you with the license. Okay, so you can install the license in your computer and you can run the calculations. So this is for the training purpose. So if you have a decent machine, so at least machine with the Core i5 or above with at least 4 GB RAM and at least 1 GB graphics card is needed. So if you have this type of machine, you yourself can write to us. Okay, so we will help you with the license. So there would be, as a part of this course, continuously we will be discussing various modules, so you can try out yourself. And if you are facing any difficulty, we will be having discussion sessions. So next week onwards, so once in every 3 days, we will be having QA sessions, so which will not be part of this regular series. It would be in the afternoon session, so we will be communicating it with you. So anybody having any questions, any problems, issues during those QA sessions, you would be able to ask us. So a scientist from Schrödinger would be coming online, and you can have one-on-one discussion with them, so they will help you. Further, if you register in our website, you will also have access to lot of training materials like tutorials, videos, and lot of seminar contents are there. You will get the access for that. So suppose you do not have a machine. So I said minimum requirement is any operating system. It could be Windows, Linux, or Mac machine, but at least Core i5 and above is required processor, and at least 4 GB RAM is needed. Okay, and 1 GB graphics card is needed. If you don't have that type of machine, then you can opt for a Schrödinger online course. So now this is a certificate course. So here, if you take up this, you will get a certificate. Okay, this is again here you will get a cloud-based access to the software. So this is against payment. So since we are arranging cloud provider, and also it is coming up with cost, so we are working on getting a heavily discounted price. So we are working on it. So my colleague Shelvia should be able to get the details, and she would be able to help you with the pricing detail and all. But we are working on getting a heavily discounted pricing on this. So with this, you will get by sitting at home, you can have any type of machine. You just need to have a computer, and you need to have a decent internet connection. So you would be accessing the Schrödinger software through cloud. You would be able to open the user interface, and you would be able to run the calculation. Okay, so once you finish, there are different chapters there. So you would have different stages of learning there. Even if you are very novice, you have never worked with any of the computational modeling tools, still you can go for it, because it starts from scratch, starting from sketching of molecule. So it is as simple like that. It is very simple course. So from there it will start, and it would with every chapter the complexity increases. So you can opt for that course. So once you complete the course, you will get a certificate which you can put in LinkedIn, or you can put it in your CV, which will be value addition for your CV. So for all these details, you can write to my colleague Shelvia, or you can write to ddcourse@schrodinger.com. So rather writing to Shelvia, you can write to DD course, the email which you received saying all the communication about the course we are doing through ddcourse@schrodinger.com. So you can use the same email ID to respond to us. So if you are facing any difficulty, please do not have hesitation to reach out to us. We will be there to support you. So if there is any question, I'll be happy to take up you.
Thank you, Dr. Pritesh, for that informative session. I think we received a lot of appreciation and praise for your session. And I particularly really liked your analogy with the lion, and yeah, it was really funny. So we have received a few questions. I will read them out to you. The first question is, what is the difference between reverse docking and docking?
What is reverse docking? You are trying to find out if some other molecule, or let us say, I have never heard about reverse docking. I have to dig out. Probably I should not answer at this stage. I'll get the information, then I'll say. I should not presume anything.
Right, and I think we have a separate session for docking, if I'm not wrong. All right. So the next question is, sir, can we use structure and ligand-based drug discovery simultaneously?
Yes. So if you have the information on already reported ligands, okay, let's say I'm working on a very old target. Let us say something like dihydrofolate reductase. Now, this is a very old target. So many people have been working on. So even before they got the structural information on the dihydrofolate reductase, people were doing research. They synthesized a molecule, they isolated this enzyme. Okay, isolation means you are collecting the enzyme. You don't have the structural information of enzyme, but you have collected and you are doing inhibitory activity on that enzyme. Okay, so like that you have the activity for the molecule. Further, now you can use those ligands for generating initial data. So you can go with pharmacophore or QSAR kind of analysis to identify initial virtual screening. Then whatever molecule you get as a good hits from these ligand-based drug discovery methods, you can go for structure-based drug discovery method. You can dock only few molecules which are predicted to be very high potent molecule from the ligand-based drug discovery methods. You can go for docking, and then you can run the molecular dynamics to check their binding stability. That is how you can use this information.
Right. The next question is, which approach should we use for drug discovery? Is shape-based screening is the best approach?
Okay. So in case of anyhow, tomorrow onwards, we would be discussing a lot on the detailing of these. Probably you would get automatically the answers if you attend all our session. But let me tell you, if you have maximum information about the target, that means structure-based drug discovery. So always structure-based drug discovery approaches would give you more information when you are designing an inhibitor molecule. Okay, you want to design a potent molecule, then always the molecule has to bind to the protein. That means you have to have the information of the binding site. Okay, so structure-based drug discovery tools will make utilization of the structural information of the binding site. So always these approach would give you better information. Now shape-based approach uses only the ligands information. That is only half part, and that is only complementing part you are using. You are not using the actual image, rather it is the reflection you are using. Ligand is a reflection, right? Ligand has to complement. Probably tomorrow when we start discussion detail on the structure-based drug discovery, we will be understanding all these aspects in a more detailed way. We'll be taking up. So you shouldn't be having these type of confusions. So, but shape-based, now if you compare, now what is the advantage with the ligand-based drug discovery methods? Like either shape-based, you consider, or pharmacophore-based approach if you consider. These are very fast methods. Now structure-based methodologies are not as quick or as fast as ligand-based approaches. So that is where, so for initial screening, people will use the ligand-based drug discovery approaches like shape-based or pharmacophore-based approach. Then they will use for, then they will take up the structure-based drug discovery approaches or docking approaches to finalize the molecule. Yeah. Next question, please.
Right. The next question is there a standard set of criteria you check for selecting a ligand, or is it highly specific to the structure of your target?
When we are discussing the details on structure-based drug discovery, docking or pharmacophore, we will be taking up that. So that time we will discuss all these aspects.
So there is a general question about the limitation of CADD.
Okay, every methodology has a limitation, right? Every method fails, right? Even animal models, preclinical models, you get fantastic activity, but still that fails in clinical trial, right? So every method is a representation. Unless you test yourself, I should put it, unless you test it in humans, unless you try a clinical trial, everything is a model, and every model has a limitation. So it depends on the approach what you are following. Okay, with the complexity, with the approach, the limitation of each application, you would be able to appreciate. Probably you should attend our full course, then you would be understanding. So what is the limitation with docking based method? What is the limitation with pharmacophore based method? So all those things, each methodology will have its own limitation. So with that, how to tackle that limitation, you would be able to understand, and that's how the approaches goes. So generally when we go for, let us say in case of docking, okay, when we are taking protein, we are taking a protein and we are putting the ligand with the protein. So it is a microenvironment analysis. You are not really considering other ADME profile. Now the protein is inside the cell. whether the molecule is reaching, does it has an approach, or is it able to even reach out the protein? Those factors we are not considering. We are directly putting in the protein and we are taking. If you are going with the rigid based docking methodology, you are not considering the protein flexibility, right? So if you consider induced fit docking, then you are considering the local conformational changes in the protein. If you go with the molecular dynamics, then you are considering the local microenvironment, but only protein you are considering, not other ADME profile you are considering. So every technique will have their own limitations. So once you understand the applications or the approaches, then you would be able to work on overcoming those issues.
Right. I think you already touched upon a little bit on the next question. So it is, what approach is best, flexible docking or rigid docking?
It all depends on the scenario, what type of scenario you are going, which stage of drug discovery you are starting with. Now, in case of, I'll give you a very simple example with COVID scenario. In the COVID scenario, we don't have any information on any of the ligand. Okay. So in such cases, I have to quickly come up with the molecule. I have to identify a more meaningful molecule. So what I'll do, I'll reduce the complexities so that I can go with very fast methodology. So I can, in fact, I can start with virtual screening. Okay, virtual screening. So structure-based virtual screening. If you have some ligand information, at least you can start with ligand-based approach. But here we don't have ligands information, so we have to go with the structure-based screening. So there I would consider the rigid protein. So initial millions of molecule I'll screen, I'll identify the best molecule. Top 1 or 2% molecule I'll go for next complexity. So I'll consider then local flexibility, induced fit docking. So only few molecule I'll consider. Then few still maybe 1% of whatever molecule I consider, I'll consider whole protein's flexibility. I'll run the molecular dynamics. So it all depends on what scenario you are starting your problem with. You can decide on what type of approach would be really helpful. The time spent on a particular technique. Okay, let's, for example, so I guess it is too early to tell these points. Probably once we have the detailed discussion on different application, you would understand. Now, as I mentioned, pharmacophore screening. In 1 second, you would be able to run screening like 250, 300 molecule you would be able to screen. If you go with the shape screening, in 1 second, 10,000 to 15,000 molecule you would be able to screen. But if you take structure-based virtual screening, high throughput virtual screening, docking-based screening, for docking one molecule, it would take 1 second. So it depends on what type of technology and what type of starting material, starting point or starting position you are taking. So from there, we proceed.
Right. The next question. Can a patent be applied to the drugs discovered through computational method?
Yes, of course. If you do the ideation, if you are generating the idea, and if you are the owner of the idea or the molecule, you can file the patent.
Right. What should be the approach to find a completely new target? Sorry, the question is, what should be the approach for a completely new target?
De novo drug design, virtual screening approach. So you have to wait for 2 to 3 more days. We will take up in detail all these discussions.
Right. What could be the suggested method to screen a known phytochemical molecule to a known substrate if we can change the structure of the ligand to get more selectivity towards target? How we can know that the same ligand can't activate other targets?
There are multiple.
Yeah, I think one is about the selectivity.
Yes.
Yeah.
You need to patiently be with us. We will be touching upon all these aspects in future coming discussions. So when we talk on docking, when we talk on hydration site thermodynamics, when we talk on designing molecule, when we talk on virtual screening, when we will be talking dedicated session on database screening. All these aspects, each and every point in your question would be answered. So please attend the future discussion. For your every question, you will get the answer.
Right. All right. So when taking the target structure, which one we should select? Active form or inactive? Drug-bound structure?
Tomorrow we will discuss on that. Tomorrow I'll be talking on protein selection, right? Tomorrow we will be having a session on protein selection and all. So we will discuss on that as well. Sorry, today we will have the session.
Yeah, the second session.
Next. Miss Vinod would be, my colleague Vinod would be talking. He would be sharing these information. Just bear with us for another half an hour to 45 minutes.
Right. I think we are close to 11. So we have a few announcements. So the email Dr. Pritesh already told you. For any queries, please write us on ddcourse@schrodinger.com. You can see that on the screen. The second is a lot of people are asking about the certificates. So you will be given certificate and we will be updating you how you will be getting the certificates. But for that, you will be needing to register on our course. So please visit our website. If you have already not registered, please register. There is no specific attendance. So a lot of you are asking me if how we are taking the attendance. So there is no specific attendance, but we require you to attend all these seminars that we have live so that you can ask your questions. We will be having separate Q&A sessions, but it's always better if you are attending it live so that you can ask your question right then. Some of you are asking about the hardware, and if you do not have the hardware, can you take this course? Yes, I think already Dr. Pritesh touched upon that. So you do not need the hardware to attend this course, but you will require the optimal or the minimum configuration that we have mentioned in the form if you want to run the software on your computer. So if you'd like to go for the evaluation license, which will be a part of the phase 3 of this program. So for that, you will require the minimum hardware. But if you do not have that, you can still go for our online course. So we are looking at how to get you the best price, the discounted price for that. So please, keep checking back with us. But here is the website. If you go to our link on our website, schrodinger.com/schrodinger-online-learning, you can read up about the course and you can find out what are the features and the advantages of the course. So I think that's it for now. Please stay back. We have another session at 11:15. Till then, you can take a little break.
Shall we? I think we can start immediately rather taking a break.
Sure.
Yeah.
All right. So we can go to our next session, target structure understanding. Dr. Vinod, are you there?
Yeah.
Okay.
Am I audible?
Yes, you're audible.
Yeah, thank you very much. And let me try to share my screen.
Right, I'll just stop sharing.
Is it visible?
Yes, I see I think the entire desktop. Yeah, it's fine.
Great. So feel free to interrupt me in case if there is any audio interruption or if you're not able to hear anything. Okay. So I'm all set. If you allow me, I can get started with it.
Yes, please.
Yeah. Thank you, Shelvia. And the second topic is continuation of first introduction given by Dr. Pritesh. It is about target structure understanding, do's and don'ts. The complete slide deck is based upon my experience. This is general mistakes which is commenced by various students. Just to get started with the protein and target introduction, it's a simple analogy for those who are totally new to them. It will be a little interesting. I think every day in our breakfast, many of the participants and us have egg, and we boil our egg. And if you see, like once the egg is boiled and in raw form, it's totally different. And if I take a tentative measurement of the egg, it's approximately 10 centimeter. And if you go in a little bit deep, it's around 10 nanometer, where we end up seeing the protein all viewing. This is approximately 10 nanometers. So what is the analogy here is to analyze like why a protein forms a solid form. I would like to show you an interesting video. If you see the protein folding and unfolding scenarios, so this is what happens. So if you see, this is a protein folding. And you can see clearly like how it folds to a proper secondary structure. And here in this case, you can see how it has reached to a global minima or possible local minimum. Now what happens is similarly, like when an egg boils, the protein get denatured and it ends up forming a solid lump. I think the similar situation, many of us may forget by like cooking Maggie, and we'll forget. I have done it. So I'll forget it, and if you just leave it for very long time, it will end up in a lump, which is maybe not edible. So that's how a protein and target works out. So usually in drug discovery, the target is a receptor or a protein or an enzyme. There are many reasons for drug discovery failures. One among them is obviously poor potency or PK/PD parameters or toxicity issue. And so this is around 44% of the data represent that. And there are other reasons like around 34% of the preclinical projects don't make it to the clinical trial. There's poor documentations or lack of funding grants. So what my focus is currently now is just to focus on this target selection. Means how you need to select a commercial target. Irrespective of your domain, it is very, very important that you select a proper commercial target. And what are the challenges you face? What are the things that you need to take care? Whatever the target that you have selected, is it commercial or not? So how do you know about it? So those things I'll try to discuss here as I go along. If you're interested in getting more details upon this, there are citations in the bottom. You can go through the article later. There's another review which I liked a lot. It's like they have done a consensus study on frequently studied targets. See, commercial targets are really tricky, and major focus is mostly on GPCRs, where still date, like many of us use ligand-based drug discovery, and you have nuclear receptors and ion channels. If you see this major three chunks, it's like obviously many of them use ligand-based drug discovery. Whereas in kinases, like structure-based, because there are a dominant amount of data that are available, so structure-based drug discovery approaches are used here. And you also need to be in trend. You can't work on a very outdated target that are currently not in trend. Here is a data, this is the complete plot, and it is published in a review paper. And here it is, like it's all data is about before 1990 and after that, that is it is updated until 2015. If you notice, this orange color are the recent ones. So you can clearly see it's like immunosuppressants, anti-neoplastic agents, direct acting antivirals. So this will be very hot if you see the recent data. Otherwise, overall summary is that it is majorly life science diseases like diabetic, cancer, and so on. So lifestyle diseases are very, very commercial in nature. Now, what are the challenges of research scholars? Okay, so this all point is, I doesn't mean to say, but these are the things which I have seen, the challenges by research scholars. So one of the important is they usually get hired or they get that position due to a grant project. A grant project has already a predefined objective. If the objective is predefined, the protocol may be already set. So obviously you need to just execute your project. But it is my request, just if you are not satisfied with your grant project, you need to fulfill your project. But along with that, you need to do mini projects to gain experience on commercial targets. The other problem is experience. I think many of the PG youth scholars lack that experience as how to handle data. So it is my humble request that you need to get in touch with expertise in the domain. You need to talk with them in the conferences, get their views and opinion. So don't be too late. So try to interact with all the experienced scientists available in and around. And what happens now, again, as a student, my academic milestone matters a lot. Obviously, it matters a lot. And there is a time bound frame like, okay, I have to finish my PG, I have to finish my PhD. PhD goes pretty long. So obviously, this academic milestone also hinder the research scholar thinking ability. And guide dependence. You know, many of us make this mistake. I will say yes, guides are very supportive and very encouraging many times, but research scholars end up being highly dependent with the guide. So whatever the instruction guide gives us, we try to follow it blindly. It is very important that you need to have strong discussion with your guide on various hypotheses that you are trying to test. The next, as I said, time dependence. Like, see, it may be your PG project, which will be like maybe for second year project work. So 1 year you have duration, and PhD maybe for 4 year, 5 year. So you don't know like how quickly you are going to finish your project. And domain, like still date, it's nice to see in some private university that there is a combination of life science streams having domains like computer science and biology and mathematics and physics. So like that, they have created hybrid courses. So don't stick to your own domain. For example, if somebody has done a major with biochemistry, don't stick and wait for the opportunity that you need to do your higher studies with the same domain. I don't know. I think it's really outdated due to the requirements that are popping up now. So you need to gain expertise in multiple domains. Yes, given an opportunity, I'll request you not to fear to change your domain. Okay, the supporting research, as I said, in when you know many of the cases, the computational works are already, the experiments are already done, and they seek for some computational studies. The supporting research are quite biased in nature, where they try to prove that already available data, these are the experimental data, or let me try to support. So don't, you have to finish that task, but try to see an analogy how this would have been done using rational approaches in computations. So that is our thing. And obviously the funding problems. So many of the research scholars have troubles in getting proper fundings due to like various opportunities, or they have to write some good projects. So that these are the various challenges as per me, what I have seen. I may be wrong. And but what industry needs, industry is very merciless, like if you see. But what they need is, when you are working on, as I said, you need to have a proper therapeutic outcome. The target should be commercial for sure. You need to also analyze what are the existing IPs available for your target. What are the competitor companies that are working on? Okay, so do you have the existing molecules DMPK profiling? what are the novel heterocyclic compounds I can explore on. So majorly chemists stick to their classical heterocyclic molecule, and they don't come beyond any new heterocyclic molecule. So it is important that you work on novel heterocyclic molecules. What is the permeability study? Potency of target one, that means I mean to say the selectivity and specificity. So this is all that literature review that we try to. So these are the major keywords that I will focus on. What are the hit identification approaches that been used, and what is the options for hit to lead optimization. This is a change in mindset. If you follow all this, you may end up being an entrepreneur. Like I really see very less drug discovery companies coming out of PhD scholars. It's very sad to see, but yes, you can give a try if you properly place a protocol during your PhD. If you have a commercial target and if you identify some novel molecules, you can end up setting up your own company. This is my wish. Hope it is getting fulfilled. Role of computational chemistry in biology. Now, as I said, there is time bounding situations. So the computational biology and chemistry is going to come to the rescue. So how is it? As I said, it's a combination of multiple domains altogether. And forgive me if I have missed any important domains, but this is a collection. This is a plot to show you like how the computational biology or computational chemistry has evolved. And together with that, the advanced hardware computing has given us tremendous strength to explore so many biological and chemical scenarios using computers. It is going to save our time, effort, and cost. Okay, so what you need to do is solve your problems using computers, simulation and theory and experiment. You need to benchmark all of them and create a proper protocol, means proper understanding between the complete work. Means what are the experimental approaches available, what is in theory, and how to carry out the simulation. This is a very interesting plot where we try to analyze based upon the length scale. So what are the time scales options available, and what are the techniques that are being currently used. Now, very famous is quantum mechanics, where we try to study the electronic level effects, and then you have molecular dynamics. So these topics will be covered in the future sessions, but keep an eye on this. And then you have discrete dislocation dynamics, quantum theories. But now if you notice, and this is in very close, I mean, I don't say that it's very equivalent, but very close to the experiment that we carry out today. So you need to note down what are the drawbacks and how these computational studies are able to replicate your experimental outcomes. Don't try to have a biased approach. It is going to ruin our career. Yes, that's a nice shortcut to have, but try to simultaneously explore what are the rationale behind this experimental approach and this in silico approaches. So just to see on the rationale of CADD techniques approaches, let me just show you. This plot was already shown by Pritesh, and he was explaining you there are 4 quadrants for your selection. I have seen many of the researchers do this mistake. Okay, I want to do molecular docking. I want to do shape screening. Don't get biased with your techniques. The technique doesn't decide your research outcomes. The literature review and other analysis helps you to identify which approach you need to take. So I'll give you a small brief view. When you have targets, you need to fall into quadrant 2 and 4. Means you need to carry out molecular docking or ligand contact analysis, e-pharmacophore, or de novo virtual screening, or fragment-based drug designing, and so on. But so how do you select this? Don't get biased. So I'll just have a very nice slide. Like always, our seniors or our supervisor say, okay, go and perform your review. Okay, so we have bundle of research articles. When I initially started my literature review, it was one of the very boring thing which I don't want to do. The obvious reason is what I found out, don't try to read the complete literature. At first go, you need to classify and categorize them. This is just a sample numbers. For example, for my target of my study, there are close to 265 articles already available. Biologically, it may be a PDB or some expression study, there are 20 research articles available. Then in dry lab, this is wet lab and this is dry lab, where they have done some computational studies like structure-based drug discovery, ligand-based drug discovery, no, and wet lab and dry lab in combination, there are some research articles. So this classification is going to help you with the possibilities of finding you in one among this quadrant. For example, now I have a luxury of so many chemicals. So I would love to go into this quadrant rather than, yes, obviously I have option to explore in other quadrant because I have some receptor targets too. So don't get prefixed, and no technique is superior or inferior. It is your responsibility to benchmark and see which technique is performing better enrichment or which is able to reproduce the research outcome properly. If it's already pre-decided, why you need to take the software and do the study? It is not required, right? We would have automated it. Each research and each target is very, very tricky. So it's your responsibility to explore all the possible options. And being in PhD without the industrial pressure, you will be at the right place to do all these tests and studies. Okay, so one more problem, what happens, all the research are positively reported. I think all of or many of you may agree that the negative reporting is like, all targets are promising, everything is very nice. Then how do I know whether my target is good or bad? So only literature review, don't be just only dependent on literature review. So one thing I noticed, this clinicaltrials.gov website, it's a very nice website where this outcome decide many fall or raising of big companies. So here failures are reported. So if a molecule fails in a clinical trial, so the complete data is reported here. My request is, please cross-check with the recent 5 years targets and take a proper commercial target. If you're not going to select a challenging target, your work won't be appreciated, your research won't be published quickly. I've seen many research reviews where they say your target is outdated. There are already many reported molecules. At the end of the day, we invest our time to make our career better. So it is my humble request, please select proper commercial targets. Now, when it comes to CADD approaches, I think Pritesh already has explained you, this is like an overall workflow. So whatever I have shown you earlier, you could notice once the target is identified, the available options are structure-based or ligand-based, where you can do pharmacophore, virtual screening, lead optimization, and this is back and forth process. So nothing stops, and you can't be biased on one technique. You need to use combination of hybrid techniques to get a proper outcome. Okay. So my topic, so these are the 4 protocols. Already Pritesh just explained you on the clinical trial part: target identification and validation, hit discovery, hit to lead, and lead optimization and development candidate. But now I'll just cover the first quadrant. In Schrödinger, you can assess the available structures by using protein reliability report, visualize the electron density for any missing loops or any missing chains. So all that can be done by using Schrödinger suite. And other than that, you can do accurate protein structure prediction and refinement, loop filling, and molecular dynamics, homology modeling using multiple template, and other thing is binding site characterization. So there are high-end tools like WaterMap, and sitemap is a very handy tool, very quick tool. You have mixed solvation MD approach and induced fit docking if required. Okay, and other things that you need to also make a notice. So what are the template and homologous available for this protein? So this is going to help you to strategize properly for your selectivity and specificity study. Like it's not just one target that you need to focus on. Also, there will be a family of different protein that you don't have to bind to. So it is very important you analyze your template based upon the sequence view or domain view, like what are the domains in common. So gene view. So this all options need to be explored before getting into anything. Now if you notice, like means I mean to say that you have to analyze your complete pathway, keeping all this point into consideration. Please, it is requested that, because at the end of the day, the pathway, the whatever the recent pathway that are reported, you need to follow that. So this is one of my favorite topics, that is rational selection of your PDB. So I see still date many students make the mistake of selecting any random article, and whatever the PDB that is present there, they select that PDB without any questions. So how do you select a protein PDB from a protein data bank? The protein data bank is an excellent resource. This is an old snapshot. Now you may have higher data available. There are so many PDB deposited, and now the focus is COVID, right? So many of them are searching for COVID targets. So all the details are available. The first thing that you need to look into, like when you give your keyword search, please focus on organism. So whether the organism is Homo sapiens or mus plus or BOS or E. coli or some other enzyme. So you have to be very careful on which, but majorly many of the research focus on Homo sapiens also. But please keep in mind the veterinary medicine is also a nice industry to focus on. So think about that. Don't just stick to Homo sapiens alone. If you are thinking for little diversity, or if you feel your molecule is little toxic, you can think about a lower dosage with veterinary products too. Okay. So these are the major organisms that are most of the proteins are reported with, and the experiment. So I think many of us know in CADD, very, very critical thing is the X-ray structures are deposited, NMR, electron microscopy structures are reported, and the very recent one is cryo-EM. The cryo-EM is a very latest technology, and very limited institute have that access. So what you need to focus here is, majorly the focus is X-ray structures, which resolution less than, it's a preferred value. It's not a hard and fast rule, preferred less than 2.5 Armstrong, where the R value is equal to R free. It's a validation, and no mutation. So we call protein with no mutation as wild type. This all matrices are for preference. Okay, so in this case you can see clearly the mutation is reported, and what are the experimental data? It's a X-ray structure with resolution 2.80 Armstrong. I said less than. So now you need to be very careful in selecting your PDB. Also, please note down that the UniProt is going to give you the overall view of all the available structures. You know, all the PDBs. If you want to know all the PDBs that are reported against that particular target, so you need to go to the UniProt, and there you get the overall summary of all the PDBs. So among this, you need to select the best PDB, and there they use cross-docking technique or reverse docking. Means docking one molecule with the same family of proteins and seeing how reproducible it is. So we have a tool called as cross-docking where you can perform this docking. Okay, so that is done majorly for your same series. Now I have problem of many. I have so many PDBs. Now I can select all the PDBs, align each among them, and then I need to dock the native molecule one on the other. So the protein that is able to accommodate the major of the molecules is the best molecule for my virtual screening. And that concept is called as cross-docking or reverse docking. So what I prefer is majorly co-crystal structure. But yes, if you have a apoprotein, then you need to perform binding site analysis using sitemap. And please note down, I think many of us are not clear what is a hit molecule. Hit molecule is a molecule that has reported with single micromolar biological outcomes. Okay, you have to cross check with the literature what is the bound molecule, whether it is hit molecule or a lead molecule. So lead molecule is single digit nanomolar biological outcome, nanomolar concentration. And now what happens, see, if a molecule, a bound ligand has a lead like capability, if the reported molecule is in nanomolar concentration, then you can get an idea that it will have a better binding than hit molecule, right? When you have problem of many, like in the previous slide I showed you, like here, if you have problem of many, you can see all the bound ligands, and the ligand that has the least, that is has the best biological outcome can also be selected. So this is how the biological in the PDB it is reported. You can see this is in picomolar. Though the reporting is in nanomolar, you can see clearly this molecule has an activity close to picomolar. The receptor or the enzyme sites. Now this is another thing that you need to focus, that protein has various sites like protein-protein interface sites. Active sites, allosteric site, organic druggable site, shallow binding site, or you have to see the covalent binders also. So this all you need to clearly understand, and then approach for your docking or virtual screening and so on. Okay. Now, when you see a protein, like this is how it looks. This is the surface view. And when I see this is an organic, you can see like how this region is well defined, where yellow region is a hydrophobic region, H bond, donor and acceptor regions. So this is for organic molecules, and this is shallow binding site. So you can clearly see the difference between this image and this image. Like, because obviously any protein will try to come and hug this protein, right? So that's the reason the organic molecule binds with this site, and if a protein peptide has to bind, it will bind in this site, in a bigger site. So you can't have similar approach on both the situation. We also have a very nice product called as PLD, which is equivalent to protein data bank, where we have a lot of proprietary data also. And this is a well curated one. I will just give a brief on. It's a central data repository with deep integration into Mastro. It is very lightweight interface. This is specifically for commercial who would like to have all this data. You can pipeline this, and automatically the results of prepared protein results are already available. And proprietary software also you can customize. And the idea is, like, if you notice, the PDB database is taken, and all the other proprietary data is taken, and the crystal details, the metadata are curated in a proper searchable form. So the proprietary data that are available are crystal structure, NMR, density data, docking, models, simulation data. So all these are indexed and made searchable. That is the benefit with PLDB. If anybody is interested, this is just for information. So what are the kind of search it performs? You can mention the hydrogen bonds. You can mention the pi-pi interaction. You can mention the halogen bonds, salt bridges, metal ligand protein bonds, and do the search. So these are powerful geometric distances also you can mention, because what happens, whatever the computational outcomes or prediction does, whether it is available in crystal scenario or not, we need to cross check. And you also need to know whether this kind of nature, this kind of defined interaction is already available or not. This is going to help us to have a better foundation in selection of your residues. You can also search the molecule based upon gatekeeper residues, catalytic residues, C helix, DFG motif, hinge region, activation loop, P loop, and HRD. So these are like complex searches that are performed only in PLDB. This is not possible with your PDB searches. Okay. So this has a complete repository. I'll just show you a small images like how it is searched. Can you see here the optional wildcard, any atoms are given and what angle it should be. So once you define that, it will search. So we need to draw the ligand or the receptor mode, means whether are you trying to search for residues, are you trying to search for ligand, so those four molecules. Then you can mention the distance. Like here it is greater than 4 Armstrong. For the centroid or vector and plane objects, water and protein residues, and so on. So once the hit is done, you can also search by all the regular keywords that you use in the protein PDB. Other than that, you can mention there are filters for mutations, non-mutation, X-ray structure or model structures. So what is the resolution, what is the deposition dates, all those things you can search. So this is a simple search for a molecule where you can see here, this is R1. So any atom can come and which residue it is going to interact. So what are the other fragments you can place. So, so all these options is going to help you a proper query which is be experimental. So we need to respect what kind of experimental outcomes are available, and those searches will end up giving us various outcomes. Like for example, here you can see the hydrogen bonds, the annotations, the gatekeeper residues options. So all is set, and this is how the outcome looks like. Once the result comes, it is going to, this is the customized distance and angle between the receptor and ligand that has been mentioned. And those things will be reported in a complete chart. And this you can export as a CSV, check with Canvas, you can import in your project table. The visualizations are very, very nice. And you also get full-fledged chart report on how all these molecules are there. A complete 3D visualization in Mastro is available. So all those helps us in proper selection of PDB. So I think, so this is the view you get in 3D. And you can get an align and overall Mastro, how it looks like. This is with the kinase domain. So what kind of fragments are available. You can import your experimental data and map it. So this is a search panel, which I just showed you. And it is available for your Python API also. You can search it and integrate it in your programming language too. So you have to log in. This is the login options, and this is how the result comes. So that's all from me. I think I'll be open for any questions.
Yes, so we received a few questions.
Yes.
So the first question is, what are the characteristics of a good lead molecule?
Okay, see, lead molecule, as I clearly said, it should have a good biological experimental outcome. For example, in single digit, nanomolar or less. Means it can have a picomolar activity or nanomolar activity. So that is the characteristic of a, see, many of them misinterpret this, whether in silico outcomes are a lead or hit. No, it is biological. Okay. Yeah, thank you.
Right. There's also a question about the difference between hit and lead molecules. So maybe you can say a few lines about that as well.
Yeah. So as I say, it's just the unit of your inhibition. You know, if the molecule is in micromolar, it is called as a hit molecule. If it is in nanomolar, it is called as a lead molecule. That's all.
Right. If the X-ray structure is not available, can cryo-EM structure be used? And
You
are
need to
there any?
refine it. There is a complete new protocol being developed. So you can use cryo-EM structure, but as you know, the flexibility would be high. So you need to refine it. Currently, Schrödinger is collaborated. I don't have the correct exact details, but that research is on. But personally, you need to use it with caution.
Yeah, so maybe can you elucidate what are the few limitations of cryo-EM structure? That is another question.
As I said, this is a flexible structures. Obviously, if I am in your case, I will try to validate with a proper enrichment, and I will check the flexibility of the molecule as it is a liquid structure, right? You don't know whether the interactions are retained. Can I rely on that? So those things I will take precaution on.
Right. The next question is, what are the parameters of binding site for a small molecules to be specific to the target?
Okay, see, now as you see that in RCSB, if you see, they report the available interaction too. So in protein data bank, you cross check what your native molecule interactions are. And that is your answer for your new molecules. So that can be a guess state where you can get started with your selection or rejection. So there is no hard and fast rule. Sometimes you need to create your hypothesis in multiple ways where you select the molecules from each batch. For example, in one case, I'll just select molecule based upon binding energy alone. In other case, I'll select based upon interaction alone. I think this all will be discussed in the molecular docking session separately by my colleagues. So please, you need to hold on for some time.
The next question is, what would be the primary approach for using PLDB?
Okay, so you need to check with Shelvia. Maybe I don't know whether evaluations options are available. Shelvia, you need to give them details.
Right. So if you are interested in using PLDB, you can write to me and I'll see if we can get an evaluation license for that because it is a separate license. Okay. I think you also a little bit touched upon what is reverse docking in your presentation. And
Yes.
since somebody had asked about reverse docking, difference between reverse docking and conventional docking, maybe you can go over that again.
Yeah. Okay, so you want me to go. Okay, I'll just hold on. I'll just see if one of my papers, I will show that. And okay, so it is nothing but see what happens. This is the problem of many. Okay, so when I have collection of like 10, 20 proteins, means 10, 20 PDB-IDs, and there are different ligand bound to it. Now the problem happens like, you don't know which PDB to select. Okay, so in that case, and now all have nanomolar bound molecules or all of them have good affinity and so on. So in that case, what they do is they will align the complete molecule based upon the backbone, all these 10 molecules, and then dock one on each other in the same site. Now all the XYZ coordinate is same, right? And different binding patterns of the receptor is caught. Like obviously like a human being, you don't have a single global minima, right? While sleeping also, we have different conformations. Okay, different people have different styles of sleeping. So what happens now? You need to get a binding site that is going to identify, means that is going to allow all this molecule to produce a proper RMSD. So there is a separate paper that we have worked on it. Maybe I will place the citation. If anybody is interested, please write to me or to Shelvia. I will share that citation. So there we overall create an average of RMSD, and the protein that is able to repredict the conformation, that is biological active poses, is the best protein. And this concept is called as cross docking or reverse docking. Is it fine?
Right. Right. Yes.
And this is also can be used for your selectivity and specificity also. But the requirement is all the binding site amino acids should stay same. Is it
Okay.
okay?
Yes. So moving on to the next question. What is the importance of resolution in a protein structure and why is it important?
See, obviously, like when you take your selfie and the cameras that are available, you want better resolution, right? So lesser the value conveys the quality of your structure. If you have missing side chains, missing backbone, it's a headache for us. So if you have means that doesn't mean that you need to be too greedy on your lower values. So the ideal is like, again, it is a preferred value. It's not a hard and fast rule, because next question comes, no, I don't have 2.5, I have only 3 Armstrong, means you need to be careful. You need to check what are the artifacts with your structure, and accordingly you need to work with.
Right. So there have been a request for you to give a demo of selecting a specific PDB ID from the database. If
Time permits, I can try.
I mean, I think I don't know if you'll be touching upon that while doing the
Yeah, I
document
think
also.
yes, maybe in the protein selection, maybe that would be taken.
Okay.
Yeah.
All right. So there is a question, what are the minimum information required for selecting for working with for working with PLDB?
Again, Shelby, it's a question for you only. I think you, right?
Yeah, I think they're also asking like how are they using it, like what are the sort
Yeah,
of
it's a
uses for
straightforward
it.
search
Yeah.
query. So if anybody is taking evaluation, we will obviously help you, and it's a very the interface is very user friendly, so you don't have to worry, okay, on searchings and all that. But just check, because we also need to check with our licensing whether the PLDB is accessible, and it's like your higher version of PDB. Actually,
PDB, yeah.
PLDB is a well curated database, and so that is a higher version. And we'll check. Whoever is interested, please write to Shelvia, and we'll get back to you on that.
Right, and also like some of the features, like you can also do the same similar thing in the PDB database as well. If we are not able to get you the license for that.
Yes.
Okay. So if X-ray structure is not available, can we use homology modeling?
Obviously, again with caution. See, as you try to compromise, means as far away you go from your experimental facts, be ready for means the outcomes. Means ultimately any project success is failure is decided upon whether you got a hit molecule or lead molecule. Now you are already seeing like you can your success chances you can reduce. Now, homology modeling, now you know your success chances is less, so you need to be double cautious in approaching studies. This is end to end. It's not just do homology modeling, do molecular dynamic study and close your project. No, I mean to say you have to benchmark your homology model structure with already available structures and select a proper enrichment model, and then you can be confident with your homology model also. And it is my request that you perform biological outcomes and see whether your approach worked or not. So nobody is a boss, like your outcome, like what is your results, research results or molecule biological outcomes is going to decide whether your technique was right or wrong. So that is the reason no expert is able to tell you which technique to use. These are all open. It's up to you to try which technique and when. Is it okay?
Yes. Okay. I think there are a couple of repeated questions here. Okay, we have some time, so okay, so you mentioned about the cutoff of 2 angstrom for the selection of PDB. So they're asking why.
Preferred.
Why
I
is
said
that?
preferred.
Yeah.
Please, note down. It's not a hard and fast rule. The preferred is 2.5 Armstrong. It's my preference. You can have a preference of 2 or you can have a preference of 1, 2. But as what I have worked with my experience, less than 2.5 is preferred. Still, there is a 3 Armstrong structure, or if I have a homology model structure, so I will still prefer that also, if I get better knowledge on my research target.
Right. So another question, sometimes we only have a particular domain of protein in PDB, and also the whole protein assembly is also available. So which one is more accurate to dock with?
As per docking is concerned, your domain itself is sufficient, but when you try to validate with higher techniques like molecular dynamics, your structure won't be properly integrated. So if you have a complete structure, obviously you can go for that, or otherwise your study will be restricted to only docking and screening.
Right. The next question is what to do if the structure of the template sequence, if the template sequence identity is less than 20%. How to validate the model?
You can clearly see, first of all, I told already that homology model structure itself need to be taken with great caution, because it is slightly moving away with your experimental outcome. When your template homology is less than 20%, it's very risky for the chances of your success will be very, very slim. Still, you can take that structure, build model, do molecular dynamics, stabilize it, and you have to means the chances is very slim of getting a hit or lead molecule. Now, if you're talking about computational biology work, it's okay. Nobody is going to question us. But if you're looking about drug discovery or getting a small molecule, then the project I will consider it as a little risky. So the 20%, your question itself has the answer. Yes, you want to take a risk. Yes, obviously there are many people who have taken risk and successful, but according to the current data, 20% is pretty risky.
Okay, the next question is, can Schrödinger work on like nucleic acid receptors such as DNA and RNA?
Yes, it can work on, maybe in the demo session we'll try to cover, and but the expertise of Schrödinger software is not on nucleic acids. We can handle them like, for example, if you have a group binder, or if you have intercalator, major group binder, minor group binder, we can dock it, we can perform molecular dynamics, but you can't take big structures and can't do major studies, but yes, it can be done on a partial level.
Okay, can we use Schrödinger for bio nanoparticles?
It's a really nice topic of interest. Now we have a material science suit. So you have to give this description to my colleague Sudarshan or Shelvia, and then we can exactly tell you whether it is feasible or not. Because it's an evolving study, and you can understand nanomaterials. For example, if it is a silver nanoparticle or gold nanoparticle, and you have a protein in nature, it's a mix of quantum mechanics and molecular mechanics. It's crazy outcomes. Obviously, now there is a complete separation of quantum mechanics and molecular mechanics. So this is a hybrid study, and it is still under exploration. So you should give the details, and maybe you will get to know whether it is feasible or not. But as a supporting research, still things are being done on formulation front, or we have options to build all these materials, but you need to get in touch with the material scientists to get more details.
Right. So if you're interested in that, you can write to me and I can put you in touch with our material science scientist. So I think that's it for now. I think we have covered almost all the questions. Thank you for that nice talk, Dr. Vinod, and thank you everyone for attending and submitting a few questions. If you still have questions, you can write to us on ddcourse@schrodinger.com. So make sure you come live tomorrow as well because we will be there to answer any of your questions. So it's always better to attend live. So tomorrow's session is going to be at the same time, 10 a.m. The link is going to be the same. So all right then, have a great rest of your day. Bye-bye.
Yeah, thank you, Shelvia. Thank you all for joining. See you then. Thank you. Bye-bye.